Greetings. This is Dr. Doyle, and you are viewing Module 7 on biotransformation of xenobiotics, the hydrolysis reactions. This is the first of 9 modules on xenobiotic transformation. Biotransformation is a series of enzyme-catalyzed biochemical processes that increase the polarity of xenobiotics. Most xenobiotics are nonpolar, which enhances their transmembrane absorption. Biotransformation usually makes the molecule more water-soluble, thus enabling it to be excreted in the urine or bile. In general, the process of biotransformation is a detoxification process, although sometimes it will convert a relatively non-toxic molecule into a more toxic metabolite. The material in this module and the next 8 modules is intended to supplement the information that you will find in Chapter 7 of the textbook. Biotransformation is usually described as a 2-phase process. In the first phase, the xenobiotic is converted into an activated compound, where a reactive functional group is generated. This phase of biotransformation is catalyzed by enzymes that perform hydrolysis, reduction, or oxidation reactions. Once the xenobiotic has a reactive functional group, the second phase of biotransformation, called conjugation, can occur. Phase 2 reactions do not necessarily follow Phase 1 reactions. The diagram shown here illustrates the alternative biotransformation process for the analgesic acetaminophen. A minor Phase 1 pathway catalyzed by cytochrome P450 enzymes oxidizes the drug to a reactive semiquinone intermediate. But at therapeutic dose levels, the major biotransformation pathways are the Phase 2 processes where acetaminophen is directly conjugated with glucuronic acid or a sulfonyl group without first undergoing an enzyme-catalyzed activation step. In some cases, Phase 1 reactions can occur after Phase 2. For example, the gemfibrozole glucuronide conjugate can undergo Phase 1 cytochrome P450 oxidation. So don't think of Phase 1 and Phase 2 reactions of biotransformation as indicating a serial and temporal relationship, although it usually is. Rather, you should think of the phases in terms of the overall process. That is, Phase 1 reactions generate a reactive functional group, and Phase 2 is associated with conjugation reactions. These reactions can occur independently or one after the other. The general outcome of the Phase 1 and Phase 2 reactions of biotransformation is detoxification. This is accomplished by making the xenobiotic more hydrophilic so that it can be excreted, and in many cases these reactions also generate metabolites that are less reactive than the parent compound. However, there are also many contrary examples where biotransformation yields metabolites that are more reactive with tissue targets such as proteins, lipids, and DNA, resulting in toxicity. The main repository of biotransformation activity in mammals is the liver. Most xenobiotics undergo biotransformation in the liver following intestinal absorption. The blood carrying the xenobiotics directly flows to the liver. Also, the liver has a tremendous biotransformation capacity, both in terms of quantity of Phase 1 and Phase 2 enzymes, and also in terms of the diverse repertoire of biotransforming enzymes. These enzymes are found throughout the hepatocyte, including the cytosol and in various organelles, especially the smooth endoplasmic reticulum. However, biotransformation activity is found in other organs, including the kidney, lungs, gonads, intestines, skin, and eyes, just to name a few. In fact, some biotransformation activity that is critical to the bioactivation of some xenobiotics by nitro reduction and glucuronide cleavage resides in the intestinal bacteria. The high capacity of phase 1 and phase 2 enzymes to metabolize xenobiotics is due to their low substrate specificity. For example, human CYP2D6 and CYP3A4 metabolize a majority of approved oral dose drugs. Varied biotransformation activity in different species can often account for differences in xenobiotic toxicity to those species. As an example, rats are more susceptible to coumarin toxicity than are humans, because humans efficiently detoxify this xenobiotic via a cytochrome P450 mediated hydroxylation process. This is one reason why animal toxicity studies, although crucial to the regulatory toxicity assessments, must be viewed cautiously. There are also differences in biotransformation within human populations due to genetic differences in the expression of the phase 1 and phase 2 enzymes. This can be manifest in allelic enzyme variants with altered enzyme activity, enzyme half-life, or the levels of constitutive or inducible expression of the genes for these enzymes. Non-genetic effects on biotransformation activity include interactions by other xenobiotics. For example, patients who took cisapride, which was a serotonin receptor antagonist used for treating gastroesophageal reflux disease, along with the antifungal drug ketoconazole, developed prolonged QT syndrome and ventricular tachycardia. This was caused by slow clearance of cisapride because ketoconazole inhibits cytochrome P3A4, which is the only phase 1 enzyme involved in the elimination pathway of cisapride. Drugs that are completely dependent upon a single metabolic pathway for their clearance are called victim drugs. Victim drugs, such as cisapride, have been withdrawn from the market because of their potential toxicity. This presents another important point. Decreased xenobiotic biotransformation caused by enzyme interference or poor metabolizer phenotypes can cause some people to be hypersensitive to potential toxicity. Alternatively, if the xenobiotic is bioactivated by a specific biotransforming enzyme and that individual has a genetic trait for expressing that enzyme at a high level, then they too could experience a toxic response when exposed to low levels of the xenobiotic. The same thing could happen for a non-drug interfering substance. An example would be St. John's wort, an herbal remedy that induces CYP3A4. If a person takes St. John's wort and then exposed to the hydrocarbon combustion product benzoapyrene 7,8-dihydrodiol, they are much more likely to develop a certain type of cancer. The major enzymes that catalyze the hydrolysis of xenobiotics are the carboxylesterases, the cholinesterases, and the peroxinases. We will begin our discussion of these enzymes with the carboxylesterases. These enzymes are serine esterases and are members of the large family of serine hydrolases containing over 200 enzymes, which include cholinesterases, proteinases, peptidases, and lipases. The common feature in all serine hydrolases is that the active site contains a nucleophilic serine, an acidic amino acid, usually aspartic acid or glutamic acid, and a basic amino acid, which is usually histidine. The general mechanism for a serine hydrolase is shown here, and I have also included a larger version of the schematic in the module folder. This graphic shows the enzymatic cleavage of a peptide bond but cleavage of a carboxyl ester bond would have a similar mechanism. The first step of the reaction is substrate binding to the enzyme by nucleophilic attack of the catalytic site serine on the carboxyl carbon of the substrate. The active site histidine residue is activated by a nearby acidic amino acid to extract a labile proton from the serine hydroxyl group. The proton is transferred to the nucleophilic group that is bound to the carboxyl carbon of the substrate. In this illustration, the amino nitrogen is protonated. In the case of the carboxyl ester, it would be the ester linkage oxygen. The bond between the carboxyl carbon and the protonated heteroatom is destabilized, resulting in the release of the leaving group. In the case of a carboxyl esterase reaction, the leaving group would be an alcohol. The final step of the reaction sequence is the hydrolysis of the acylated serine of the enzyme transition state to restore the initial open active site and the release of the carboxylated product. Carboxyl esterases are capable of hydrolyzing a vast array of substrates, but bind and are inhibited by organophosphate compounds. Organophosphate compounds have a phosphoester group that binds the active site serine, but is unable to undergo the final water molecule displacement of the serine phosphoester to regenerate the active enzyme. This results in an irreversible and stoichiometric inhibition, also known as suicide inhibition. Inhibition of carboxyl esterases by organophosphate compounds is a common mechanism of toxicity. This has been observed with a number of pesticides, for example, parathione, and by nerve gases like sarin and soman. A vivid and sobering reminder of how dangerous exposure to organophosphate compounds can be was demonstrated by the bioterrorist attack on Tokyo subway passengers using sarin gas. Please view the ABC News video regarding this tragedy that is linked in the Module 7 folder. Most of the carboxyl esterases are found in the endoplasmic reticulum within hepatocytes in the liver. There are some soluble carboxyl esterases, called cholinesterases, that we will discuss very soon. In the liver, the two major carboxyl esterases are simply called human carboxyl esterase 1 and human carboxyl esterase 2. The two types of carboxyl esterases can be distinguished by the leaving groups that are produced during catalysis, although some xenobiotics can undergo ester hydrolysis by either enzyme. The human carboxyl esterase 1 enzyme reacts with substrates that yield a small alcohol leaving group. An example of this is shown here with oseltamivir, which is an antiviral drug. Like many other prodrugs, it has an alcohol group attached via an ester bond to make the molecule hydrophobic so as to increase the intestinal absorption. The prodrug is activated in the liver through the action of carboxyl esterase 1. An example of carboxyl esterase 2 catalysis is shown here for cocaine biotransformation. Carboxyl esterase 2 cleaves an ester linkage yielding the ecogonine methyl ester, EME, and benzoic acid as products. As you can see, EME is a rather large alcohol leaving group, which is a characteristic for carboxyl esterase 2 catalysis. Cocaine can also be metabolized by carboxyl esterase 1, which yields the small alcohol methanol, and benzyl ecogonine, BE. Also, we can see that carboxyl esterase 1 can catalyze a transesterification reaction, if ethanol is present, removing the methyl ester and replacing it with an ethyl ester. Cholinesterases are also members of the serine hydrolase family. Therefore, the catalytic mechanism is essentially the same as the one for the carboxylesterases. There are two types of carboxylesterases: the acetylcholine esterases and the butyrylcholine esterases. Acetylcholine esterases are found in nerve and muscle tissue. In these tissues, they function to terminate the signal produced by the acetylcholine neurotransmitter. Thus, the activity of this enzyme reduces the level of acetylcholine in the neuromuscular junction of a skeletal muscle fiber and thereby diminishes the stimulus for muscle contraction. Butyrylcholine esterase is a soluble enzyme found in the plasma. Acetylcholine esterase and butyrylcholine esterase obviously catalyze the cleavage of acetylcholine and butyrylcholine, but they also cleave other ester linkages, such as those found in the drugs methylprednisone acetate, procaine, succinylcholine, tetracaine, and others. Like the carboxylesterases, both cholinesterases are inhibited by organophosphate compounds. An important point that I will make here is that the most critical target in terms of organophosphate-induced toxicity is the acetylcholine esterase enzyme. This is because of this enzyme's key role in regulating the nervous system and muscular activity. One could look at the soluble carboxylesterases and the butyrylcholine esterase, which are both in the blood, as molecular decoys that may divert some organophosphate toxicants and minimize inactivation of acetylcholine esterase. A person exposed to organophosphates, for example sarin gas, is in an acute life-threatening situation because of acute toxicity caused by respiratory failure due to paralysis of the diaphragm. Fortunately, there are antidotes for this type of poisoning, including the use of atropine to block acetylcholine receptors and the use of pralidoxime, also known as 2-PAM, to reverse the organophosphate inhibition of the acetylcholine esterase enzyme. In the next slide, we will look a little bit closer at the treatment of the patient who is poisoned by an organophosphate. Shown in the top panel is a diagram representing the interaction of an organophosphate compound with the active site of acetylcholine esterase. The phosphate group is a stronger electrophile than the carboxyl group in acetylcholine. Hence, the organophosphate forms a much stronger bond with the enzyme that is not effectively reversed by hydrolysis during the final regeneration step of the catalytic cycle. In fact, this bond to the enzyme's serine residue is further strengthened by the loss of alkyl groups from the organophosphate during a process called aging. It would take roughly 2 to 3 months for the irreversibly inhibited acetylcholine esterase to be replaced through new enzyme synthesis. The 2-PAM antidote for organophosphate poisoning is an oxime that accelerates the dephosphorylation of the enzyme. 2-PAM has good binding to the acetylcholine esterase enzyme because of its ability to bind to the anionic site that is used for the binding of the cationic choline portion of the acetylcholine ligand. This allows the oxime group, a strong nucleophile, to bond with the electrophilic organophosphate. This relieves the bonding with the serine of the acetylcholine esterase, allowing the 2-PAM organophosphate conjugate to dissociate from the enzyme. It is important that the antidote be administered as soon as possible after organophosphate exposure because the strengthening of the bond between the organophosphate and the enzyme during aging can reduce the effectiveness of 2-PAM therapy. Peroxinases are enzymes that hydrolyze the ester linkages of a very diverse group of endobiotics and xenobiotics, including organophosphates, organophosphonites, aromatic carboxylic acid esters, cyclic organic carbonates, and lactones. The name peroxinase is based on the ability of the PON1 isoform to cleave the organophosphate pesticide peroxone. The three peroxinase isoforms are all found in the liver. PON2 is found in most tissues, and only PON3 is a soluble enzyme found in plasma, although PON1 is found in blood associated with high-density lipoproteins. PON1 is unique among the liver esterases in its ability to cleave and detoxify organophosphate compounds. It is not understood why this enzyme is not irreversibly inactivated, like other esterases, but it may be due to calcium-catalyzed hydrolysis. All three isoforms have the ability to hydrolyze or form lactones, so some have suggested that these enzymes should be called lactinases. An example of this lactinase activity is the PON3 catalysis of lactinization and hydrolysis of statins. Some phase 1 processes involve oxidation of conjugated carbon-carbon double bonds in aliphatic or aromatic molecules, resulting in the production of alkene or arene epoxide structures. Epoxides are highly reactive electrophilic groups that will attack nucleophilic molecules in the cell, such as proteins and DNA, forming adducts that can change cellular function or create genetic mutations. Epoxide hydrolases are enzymes that add a molecule of water to the strained epoxide ring, generating less reactive vicinal diol groups. The epoxide hydrolases that utilize xenobiotic substrates are found in the microsomal and soluble cell fractions obtained from virtually all tissues. Microsomes are membranous structures produced from fragmented endoplasmic reticulum during the homogenization of cells and the preparation of cell fractions. The active site of these enzymes is very similar to that found in the serine hydrolases, having a catalytic triad composed of an acidic and a basic amino acid and a nucleophilic amino acid to which the substrate covalently binds in the enzyme transition state. This slide shows the structure of the catalytic site in an epoxide hydrolase from Agrobacterium radiobacter. Although this is a bacterial enzyme, the catalytic site is similar to that in humans because the epoxide hydrolase enzyme is strongly conserved during evolution. The xenobiotic substrate shown in blue is styrene oxide. As we have seen with the carboxylesterase catalytic triad, we see here that an electrophilic carbon within the epoxide ring is attacked by the nucleophilic oxygen, in this case from aspartic acid. This causes ring opening as a bond is formed between the substrate and the transition state enzyme. This is then followed by nucleophilic attack from a water molecule which was activated by proton abstraction by the nearby acidic and basic amino acids. Most of the alkene and arene epoxide substrates are generated through the action of cytochrome P450 enzymes that are located in the smooth endoplasmic reticulum. These highly reactive oxidation products may be toxic to the cell unless they are rapidly converted by epoxide hydrolase to the less reactive diol metabolite. This means that there must be a close relationship between the enzymes that produce the epoxides and the enzymes that break down the epoxide. This is accomplished in two ways. The first is that the regulation of the genes encoding the epoxide-producing cytochrome P450 enzymes and the epoxide hydrolase enzymes are coordinated. Thus, induction of the cytochrome P450 genes corresponds with an induction of epoxide hydrolase genes, with proportional increases in the proteins that are synthesized. The second way that these systems are coordinated is that the two types of enzymes are spatially located next to each other within the smooth endoplasmic reticulum. This results in the epoxide being handed off from the cytochrome P450 as it is being produced to the epoxide hydrolase, where the epoxide ring is immediately broken. As you can see, the role of epoxide hydrolase is generally a detoxifying one, although there are exceptions, as we will now see. This pathway shows how epoxide hydrolase can increase the toxicity of a xenobiotic. The parent compound here is benzo-A-pyrene, a heterocyclic aromatic hydrocarbon that is generated from combustion. This is an air pollutant that is derived from industrial smokestacks like incinerators and is also a component of cigarette smoke. You can even be exposed to this chemical by eating charcoal broiled meat. Benzo-A-pyrene undergoes a phase 1 oxidation by CYP1A1 and other cytochrome P450 enzymes, including CYP3A4. Oxidation of the arene ring can result in the formation of an arene epoxide at different positions of the pyrene molecule. The 4,5 oxide is readily detoxified to the 4,5-dihydrodiol by microsomal epoxide hydrolase. However, if the 7,8-dihydrodiol is further oxidized, an epoxide ring is generated in the 9,10 position. This is not a substrate for epoxide hydrolase because of steric hindrance from the pyrene carbon structure between the 10 and 12 positions, also known as the Bay region. This results in the sustained presence of a reactive electrophilic oxygen in the epoxide ring, which will bind to DNA molecules. Some of the DNA adducts formed may include critical gene targets such as the Harvey-Ras oncogenes. Mutations in this or other critical gene targets can result in tumorigenesis. This completes Module 7. Please test your knowledge of the material by taking the self-assessment quiz located in the Module 7 folder.